Hi, I'm Mark Horwich from Yale School of Medicine and HHMI, and I want to tell you a story about a discovery that has led to a really wonderful, what's now 15 to 20-year experience of experimentation that followed. This discovery was really somewhat serendipitous, and I think there was a lot of luck involved. It was helped enormously by two wonderful collaborators who've remained very close friends and constant contacts, Ming Cheng, who's now in Taiwan, and Ulrich Hartl, who's now in Martinsried. And so I'll just tell you the story as it occurred. So the story is about information transfer in the cell. As we know, DNA is transcribed into RNA, is in turn translated into protein. And the step that's involved here is the final step of information transfer, namely that of protein folding. So we know about machines that carry out transcription and translation and have very fine molecular details to those reactions for some years now, increasingly fine. But protein folding has remained somewhat mysterious, and it's really only during the last decade or two that we've really started to gain some understanding about how this works in cells. So for those unfamiliar, I'd just point out that proteins are composed of 20 different amino acids, and these strings of amino acids ultimately fold into characteristic three-dimensional structures that carry out biological activities. For example, they could be enzymes, cytoskeleton, hormones, hemoglobin channels, receptors, what have you. Proteins are major effectors in the cell, and so their proper folding is crucial to them being biologically active, because when they're effectively strands of spaghetti leaving a ribosome, they don't necessarily have any biological activity at that point. They have to be folded correctly in order to be active. And so the granddaddy experiment of protein folding that I heard about when I was an undergraduate at Brown University in 1972 was carried out by Christian Anfinsen. In fact, in the late 50s, he received his Nobel Prize for this work in 1972, a really astonishing experiment where he started with an enzyme, ribonuclease, 100-odd amino acids with 4 disulfide bonds, and unfolded that protein by treating it with denaturant urea and reductant mercaptoethanol. That produced essentially a random coil version of the protein, and he asked whether when he removed the urea and reductant, whether the protein could find its way back on its own in a test tube to the native active form, and astonishingly, it did so. So this proved that a polypeptide chain contains all of the information for folding into the unique native structure in its primary amino acid sequence structure. He also presumed that, in fact, this native state would lie at an energetic minimum. And so this astonishing experiment led people to believe that perhaps inside cells, proteins also fold spontaneously. But increasingly, there were instances where that didn't really happen. Both in a test tube where many proteins could also not be spontaneously refolded, as in the Anfinsen experiment with ribonuclease, but inside cells, when the biotech industry began to express mammalian proteins in E. coli, for example, they often saw these terrible-looking aggregates with no biological activity and failed to recover their favorite proteins in a native active form. And this, once again, one saw, as in the test tube, a misfolded, aggregated inclusion body, as it were. And so this suggested that something more might be required in the cell to deal with these so-called, what would be kinetic problems of folding, where proteins simply get it wrong and go off pathway and misfold and aggregate. And so we never thought we would have anything to do with this, but we were busy in the late 80s studying mitochondrial protein import. So it turns out that most of the proteins of mitochondria are encoded in the nuclear genome. The messages for these proteins are then translated on cytosolic polyribosomes and an N-terminal targeting peptide in these proteins directs them to mitochondria. And they're then imported through the mitochondrial membranes, and the targeting peptide is then clipped, and the mature sized protein then has to fold into its biologically active form. Well, an important experiment was carried out in 1986 by Gottfried Schatz and his co-workers that showed that, that a protein had to be unfolded in order to transit the mitochondrial membranes. He used dihydrofolate reductase as a substrate in these experiments, showing that if you lock DHFR into its native state by adding the ligand methotrexate, the protein couldn't go through the membranes. Whereas if you now removed methotrexate or denatured the protein, it would go through the membranes. So the question that arose to us as we were studying mitochondrial protein import was, do proteins, upon reaching the mitochondrial matrix space, refold spontaneously to their active form, as might be suggested by the Anfinsen experiments, or could there be a machinery that was required to assist the protein to reach its native active form? And so we were in a position to actually test this because we had a bank of temperature-sensitive lethal yeast mutants that contained a galactose-regulated version of a mitochondrial protein, ornithine transcarbamylase, a human protein that we had shown in yeast already could be expressed and would go faithfully to its mitochondria, have its signal peptide cleaved, and become biologically active and make OTC enzymatic activity. And so our screen was basically to start with temperature-sensitive lethal yeast mutants, the idea being from Schatz and others in the field that if you can't make new mitochondrial proteins and new mitochondria, the cell basically stops its growth and ultimately it's a lethal. So we started with a bank of lethal mutants programmed to express mitochondrial OTC behind a gal operon promoter, and we proceeded to test each mutant individually for the ability to make OTC activity after shift to non-permissive temperature and shift into galactose to turn on OTC expression under non-permissive conditions. We then analyzed OTC activity. If it wasn't present, we would then Western blot the particular mutant after shift to 37 degrees and induction of OTC to see what the fate of the OTC subunit was. Was it present as a mature size protein, or was it still a precursor that hadn't been recognized by mitochondria? And so our idea was to dissect the mitochondrial import pathway. Well, as we were asking this question about refolding inside the mitochondrial matrix, we knew what we would be looking for in the form of a mutant that affects refolding in the mitochondrial matrix space. We would have mature-sized OTC subunits because their signal peptide would be cleaved and they'd be inside the matrix space, but we would have no enzymatic activity because they would have failed to correctly fold, and in the case of OTC, trimerize into the active form of the enzyme. So we started to look through our bank of mutants, and to our astonishment, almost right away, having asked this question, we found a mutant in which exactly that happened. So OTC was our initial reporter enzyme, and what we found was that OTC subunits entered, they reached their mature size, and then there was no biological activity at all. So this astonished us. It seemed like heresy to have a result like this. So we thought, we better start to look at endogenous yeast proteins and see what is happening to them. So we looked at this particular protein, the F1 beta subunit of ATPase, of the F1 ATPase, and we found that in fact, similarly, this beta subunit had failed to assemble into this stalk-like structure. And the subunits themselves looked like they had misfolded in the mitochondrial matrix space. We were in a bit of a quandary because, again, it seemed like heresy, and we couldn't really feel comfortable about the data at hand. And we were sort of pondering what proteins to look at next when thankfully the phone rang, and it was Ulrich Hartl and Walter Neupert calling from Munich to say, well, we understand that you guys have some mutants that, of yeast that affect mitochondrial protein import, and we would love to get together with you and perhaps help on some of the biochemistry of those mutants if you'd have some interest. So at that point in history, my lab had 4 people in it, and we weren't very well versed at fractionating yeast mitochondria, whereas these were world experts. So we were just delighted to interact, and I went to Munich the next week and presented some of the better described mutants that we had that affected mitochondrial signal peptide processing. But I also, at the end of my talk, described this particular mutant. And Walter and Ulrich were somewhat worried that what was happening in this mutant was that proteins were coming in through this contact site, but weren't getting entirely into the mitochondrial matrix. That they were poking their N-terminal signal peptides in, and the signals were being cleaved, but then the proteins weren't entirely entering, and of course they wouldn't fold if they couldn't enter through the membranes. And so I said, well, I'd be absolutely delighted to send you the mutant, and you can make mitochondria, I am sure, from this mutant very effectively and analyze to see what's going on. In fact, as soon as I got back, I sent the mutant to Munich, and 2 weeks later, Ulrich called up to say excitedly, you're right, the proteins are entirely in the mitochondrial matrix. I can add exogenous protease, and it doesn't touch them. So it seems like they have gone into the matrix space and they have failed to fold correctly. So this was a really wonderful time where there was a lot of back and forth with Ulrich and I crossing the Atlantic to visit each other and discuss the phenotype of this mutant. This is us together at his parents' house. Here's his lovely wife, Monachit, who is a biochemist herself and has worked some on the GROW-EL system as well in recent times, a chaperonin of bacteria. And maybe this is her telling us how this is all going to work out. But in any case, we carried out together a set of further experiments on a number of mitochondrial matrix proteins to see what was happening to them. And one of the most exciting results came with this particular protein, the risky iron-sulfur protein. So this is a protein that is imported to the matrix space and has to be cleaved twice as a monomer before it enters the intermitochondrial membrane. And when we looked at this iron-sulfur protein, we found it in either a completely uncleaved state or in a once cleaved state, as if the protein had not folded correctly and had not undergone the proper cleavage that enables its correct biogenesis. So this was the first of a monomeric protein where we saw effects in this particular mutant. At this point, we rescued the mutant with a yeast library. So we put on a bank of yeast clones, and the gene that rescued was sequenced by Ming Cheng during, I believe it was summer 1988. And at this point, we knew that there was a modestly heat-inducible protein inside mitochondria that had been identified the year before by Richard Hallberg, working in Iowa, in Tetrahymena. And we had a feeling that Hallberg was analyzing the sequence of the corresponding homologue in yeast with a view to doing further experiments in that system. So on a Saturday afternoon in that summer, Ming Cheng called up Richard Hallberg to see whether her sequence of this gene would match the sequence that he had of the homologue he was studying. Well, I had gone to JFK Airport to pick up Ulrich because the two of us were going to a Gordon conference on mitochondria the next day. And my son came with us, and it was many hours. But we got back to our little beach house at around 6 o'clock in the evening. And there's Ming Cheng and the rest of the lab standing in our front yard. And Ming's just jumping up and down holding a sequencing gel saying, "It's a dead match, it's a dead match." And we said, "Well, what's the dead match?" She said, "Oh, I called up Hallberg. Our protein corresponds to this ring assembly that he's studying." And so that 2 and 2 came together in a really amazing moment, and we had a wonderful evening of celebration. But basically what it amounted to was what we then called heat shock protein 60, collectively with Richard and Ulrich and Walter and everybody, this modestly heat-inducible protein was actually assisting the folding of all these proteins being imported into the mitochondrial matrix. And in fact, however, HSP60 is essential at all temperatures. So its assistance to polypeptide chain folding is required under normal conditions, which embody often relatively high temperatures and very high solute concentrations in the mitochondrial matrix. Ergo, this machine is essential under all conditions. Now, shortly after this, Ulrich carried out a beautiful experiment with Joachim Ostermann, where they analyzed another monomeric protein, dihydrofolate reductase. And what they could see was that when they put a signal peptide on DHFR to make it go into mitochondria, much as in the Schatz experiments, letting it go into mitochondria, it became associated with this complex, with this ring assembly that Hallberg had first identified, the HSP60 complex, as long as ATP was relatively deprived from mitochondria. If they added ATP back to this system, they now saw DHFR depart from this complex and appear as a native folded protein. While the protein, I should say, was associated with HSP60, it seemed to be non-native, very protease susceptible, recognizable by antibodies that specifically saw non-native forms of DHFR. And so these experiments really with risky iron-sulfur protein, with DHFR, convinced us that HSP60 was some sort of a folding machine in the mitochondrial matrix. And of course, there's some generality to this because when we saw the sequence of HSP60, we knew already that it was going to be homologous and is homologous to the GRO-EL protein in the bacterial cytoplasm, essential in that context, and had been shown to be involved or had been implicated early as being involved with lambda phage head assembly. Now this idea had to move to lambda phage protein folding. There was also a protein inside of chloroplasts called the Rubisco binding protein, identified by John Ellis as being required for assembly of Rubisco, which is a CO2 fixing enzyme, in fact, the most abundant protein in the biosphere. This now had to go to the level of thinking that Rubisco binding protein was actually mediating protein folding of Rubisco subunits. And finally, shortly thereafter, working together, Ulrich and I with Jonathan Trent identified a heat shock protein in thermophilic archaebacteria that we called thermophilic factor 55, another double ring assembly that is highly induced when you take these normally heat-loving bacteria and take them to an even higher temperature, that turns out to look just like a chaperonin. And it also could turn over ATP and behave just like these other ring assemblies. And in fact, when we analyzed this subunit, we found a homology to a protein in the eukaryotic cytosol called TCP1, or CCT for short, this being the eukaryotics, a member of the eukaryotic cytosolic chaperonin, one subunit of the 8 subunit rings that comprise that chaperonin, essential for folding actin, tubulin, beta propeller proteins, and many other proteins. So the generality of these ring assemblies and mediating protein folding to the native state seem to be getting established. And so finally, mechanistically, 20 years later, we understand at least how the bacterial chaperonin system works in pretty good detail now with much biochemistry, X-ray crystallography, NMR studies, and a lot of EM conducted on this particular machinery, the GRO-EL machine and its cooperating component GRO-ES. And the bottom line in terms of mechanism is that an open ring of GRO-EL with all this hydrophobic surface specifically binds non-native proteins through their exposure of hydrophobic surface that will be buried to the interior in the native state. Thus, GRO-EL specifically recognizes non-native proteins. And once it's bound to protein, when GRO-ES now encapsulates the same ring that's bound by polypeptide, the polypeptide is released into this hydrophilic folding chamber. All this blue here signifies electrostatic residues. And the polypeptide now folds in isolation in a sequestered environment where it simply can't aggregate with any other protein. And it basically uses Anfinson's principles. Its primary structure to find its way to the native state without the fear of aggregation or going off pathway and being lost. So in general, molecular chaperones, if one wants to talk about all of the classes of these components, they basically recognize exposed hydrophobic surfaces that are proffered when a protein misfolds or during biogenesis. And the idea of the chaperone is to bind these surfaces and prevent them from self-associating with each other in multimolecular aggregates. The idea of all of the chaperones is to prevent aggregation by binding these surfaces in various geometric contexts. The remarkable thing about the chaperonins, however, that's really unique is that they use nucleotide to release the polypeptide into a sequestered folding chamber where the polypeptide really has no chance of aggregation and has a chance to fold using its own primary structure to the native state. One of nature's really most beautiful machines used for this final step of information transfer. So thanks very much for listening to this lecture. I hope you enjoyed it. Thank you.